Let's take a look at some of the key concepts that relate to metabolism altogether here. And so let's remember a couple things about metabolism. Remember that it is the sum of all of the chemical reactions that we need in the body. Remember that texts and textbooks are simply going to be discussing this as it relates to energetics and possibly genetics in relation to protein development. Energetics is discussing the issues of regenerating ATP from ADP and AMP. It's based on the laws of thermodynamics. The genetics is going to be addressed, normally is going to involve protein synthesis, and very rarely do any of the books look at issues of lipogenesis, lipolysis, amino acid synthesis, or degradation. We're going to take a look at most of these issues here as we work our way through metabolism. So just a general reminder of what metabolism happens to be in terms of the total reactions for the body. We're going to spend a lot of time focusing on this aspect of metabolism here, which is the energetics of metabolism. A couple of key concepts we have to remember when we're looking at metabolism, and we have to remember these because they're going to get interchanged. So reactants are molecules that are reacting with each other, and products are things that are being formed that will go about becoming the next thing within the pathway. Now, sometimes we'll refer to these as substrates. More often than not, I'll be using the word metabolites. Reactants that form products are metabolites. They're metabolites within the sequence of events. Sometimes we'll go about talking about as being substrates as well. The other concept that we have to get down is the concept of byproduct. And when we're talking about a byproduct, we're talking about a molecule that is being formed, but does not directly get involved with any of the other steps within the pathway. And so when we look at the total of the biochemistry within the pathways, we have to remember what is the product and then what is the byproduct. Along with that issue, we have to remember that the function of the molecule is based off of the arrangement of the functional group, and I've given you a list of all the various functional groups that we have to worry about. You're not going to be examined on them, but you do need to realize what they happen to be as we discuss them, as well as the shape of the molecule, in which we have a benzene ring, which will have a distinct function. A benzene ring is a hexagonal ring that has what's referred to as a floating double bond within it. And so this is how we diagram out the benzene ring. And we'll get more diagrams when we do our chemistry lab. Our cis arrangement, where the functional groups of the molecule are on the same side of the carbon backbone, or the trans arrangement, where the functional R groups are on opposite sides of the carbon backbone. The other thing we have to remember when we're looking at the biochemical reactions here is the issue of energies and the means by which we go about measuring the energy. Free energy is the energy that is not contained within the molecules that we are dealing with. It's energy that is either being absorbed or energy that's being released from the reactions. And activation energy is the energy that is necessary to the environment to allow for the reaction to take place. When reactions take place, they can be either unidirectional or bidirectional. And so unidirectional, A reacts with B via an enzyme to form C, or C interacts with an enzyme to break apart to form B. The other way of looking at this is with what's referred to as bidirectional, and this is where A and B will interact with each other to form C, and C will degrade back to A and B. In the unidirectional reactions, we tend to have what's referred to as a static equilibrium. That is, the enzyme will be slowed down based off of the concentrations of C being formed during the anabolic reaction or A and B being formed during the catabolic reaction. However, in bidirectional, it's usually referred to as a dynamic equilibrium. That means that we're going to make sure that the concentrations of A and B and C are in perfect harmony with each other at all times. And so we're constantly speeding up and slowing down the enzymes that are coming into play. And so how do we go about speeding up and slowing down the enzymes that are coming into play? It's all about how we regulate or inhibit enzymes. And there's a couple ways of doing this. One way of doing this is what's referred to as competitive inhibition. And this is where we have substances that have similar chemical structures will bind onto the active site within the enzyme, thereby limiting the ability for the substance that we want to react. Or through what's referred to as allosteric or stereoscopic modification. This is where we need to have a secondary molecule which will either open up allosteric activation or close off allosteric inhibition, the binding site for the enzyme. By doing the allosteric regulation, what it does is it changes the shape of the molecule, and it changes the shape of the molecule, thereby modifying the shape of the binding site. By doing that, it changes the relative affinity for the substrate to the enzyme, and thereby limiting the rate at which the enzyme can go about functioning. The last of the concepts that we have to remember here is what's referred to as the oxidation and reduction reactions, or the redox reactions. Now, oxidation and reduction has to deal with the movement of electrons, in which oxidation reactions we are going about and moving or losing electrons, whereas in reductions we're going about moving electrons so as to gain electrons. Now, it's very hard for us physiologically to see the movement of electrons. However, we can see this with the movement of hydrogens. And so a lot of times we'll say that things are in a reduced state when they have hydrogens attached to them. Because wherever the hydrogens are going to move, particularly the hydrogen ions are going to move, the electrons associated with that hydrogen nucleus will follow along. Now, in the redox reactions, in the reduction and oxidation reactions, reactions can occur at rates that do not allow for the full transferring of the electron and the proton within the hydrogen. This leads to the formation of what's referred to as the radical. Sometimes the radical is referenced as being the reactive oxidative species, ROS. And this reactive oxidative species exists in such a way that we have an electron in the valence shell that is unpaired and causes an irregular spin to take place within that valence shell. And by having this irregular spin within the valence shell, we have a molecule that wants to react and bind onto something. In doing this, it forms what's referred to as a radical, which means that it's a molecule that is attempting to bind onto something because it has an electrostatic attraction to it. In order to neutralize that radical, we have molecules that are referenced as being antioxidants. The antioxidants, it has nothing to do with oxygen whatsoever. Remember, we're talking about reactive oxidative species. And the two most prominent molecules involved with the formation of ROSs is nitrogen and oxygen. Most of the ROSs that get formed within energetic pathways. There are secondary ROSs that get formed within hepatocytes, which we'll talk about with hepatocytic metabolism. What they're attempting to do is that these radicals are attempting to pair their unpaired electron. And what it does is it causes an electrostatic shearing as the radical starts to interact with other molecules that are surrounding it. We have molecules that will act as reducers within the redox reactions to neutralize the radical. Now, it's not a permanent neutralization, it's a temporary neutralization. And so what ends up happening is that the antioxidant molecule has an extra electron, but it has an extra electron that is not impacting the valence shell spin of all of the electrons there. And what it's able to do is it's able to go about and provide a temporary binding site for this radicalized reduced agent. By doing that, it stabilizes the spin and stabilizes the radical until it can get eliminated from the body. We see a lot of these antioxidants within the mitochondria. There is discussion that we have to somehow supplement our diets with antioxidants. The problem with that is that our mitochondria and our cells are producing enough antioxidants if we are healthy to match the rate of ROS and radical development within our cells. And extra antioxidants do not actually provide the benefit that is purported to occur from consuming the antioxidants.
Enzymes enable molecules called substrates to undergo a chemical change to form new substances called products. Each enzyme acts on a specific molecule or set of molecules called substrates. Each substrate fits into an area of the enzyme called the active site. This fitting together is often compared to a lock and key mechanism. However, the enzyme changes shape a little to fit with the substrate. In the enzyme-substrate complex, the enzyme holds the substrate or substrates in a position where a reaction can occur easily. After reaction, the enzyme releases the products and can go on to carry out the same reaction again and again.